Hi, so I'm going to talk about phenotype space, which is a very important concept in evolutionary biology, and in particular about the underlying structure of phenotype space. So phenotype space for empirical studies is approximated by morphospace. So briefly, a space can be formally defined as a set of elements which is equipped with a notion of relationship among these elements. And so a morphospace, as we use them, is a multidimensional space that has axes that represent measures of morphology. Usually looks like this, and we use as shape distance as this metric to relate the different organisms. So a classical textbook example of morphospace is a rope morphospace about shell coiling. And so in that type of space, we have three parameters, and the combination of three coordinates for each of these parameters corresponds to one location in the space, and so to one possible shape. Morphospaces have been widely used in evolutionary morphology in different fields, like theoretical morphology, evolutionary ecology, disparity analysis, or functional morphology. So they're really a powerful tool. There's still sometimes problem with morphospace, and particularly regarding their interpretation. There are some methodological problems, but I'm going to talk here about a biologically grounded problem. In morphospaces, organisms are usually depicted as points, usually like adult phenotypes. But organisms are not static adult entities. They are developmental systems. So instead of point, actually, they have an ontogeny, which can be depicted as this line in the age, size, and shape space. And the pathway drawn by the taxa during its ontogeny is called the developmental or the ontogenetic trajectory. So development corresponds to all the processes that link genotype to phenotype. There's a bunch of them here. That's pleiotropy, which is illustrated, but there's canalization, epistasis, constraint, all that stuff, which make that the mapping from genetic changes to phenotypic effect is complex and nonlinear. And this has important consequences for the structure of the morphospace and also for our intuitive interpretation of the notion of distance and direction in morphospace. So I'm going to illustrate this problem with a case study which is well understood. It concerns two species of trilobites from the genus Zachanthopsis. I will refer to this pattern, like the transition to the ancestral Levis to the descendant Palmeri, as the Levis-Palmeri transition. So there are two early Cambrian trilobites, and we can characterize their shape using geometric morphometrics. So we have a set of 25 landmarks that basically capture the shape variation among these specimens. That's what it looks like with just a set of landmarks. We perform the classical geometric, like generalized Procrustes analysis to obtain a Procrustes shape space. So you have these two species. Levis is in red and Palmeri in black. The size of the dot corresponds to the size of the specimen. So you have the two ontogeny here. So you can see that they start both at the same location in the shape space, so with very similar morphologies. And then during ontogeny, they tend to diverge, ending up with quite different shapes. So the analysis of this transition has been done, and it corresponds to what we call mosaic heterochrony, which is basically you have three different modules in the cranidium, so that's the head of the part of the head of the trilobite. So three different modules that each evolved using a different type of heterochrony, and the combination generate that complex pattern at the global scale. So we have three modules evolving by heterochrony. That's going to be our set of constraints for a little theoretical experiment. So we have this shape space and the transition that would be observed between the two adult phenotypes from Levis to Palmeri. There's a distance between them, a shape distance. And there's also, from that ancestral condition, another set of possible shapes that are equally distant from that ancestral condition. So we're going to build this sort of compass with all these shapes, and we're going to look at how easy, or what's the amount of evolutionary developmental changes, using the constraint mentioned before, that we have to require, like, to reach this, or to generate these different shapes around the ancestral condition. So you have like subtle but still significant shape variation. They all correspond to different shapes because they are all at different locations in the shape space. So that's shape variation along the x and y axis. So how do we measure this amount of evolutionary developmental changes that I'm going to refer to as accessibility? Well, first we map on that plane a regular sampling grid. So each sampling point corresponds to one possible phenotype. Then we look for each shape. We can decompose it into its three constitutive modules, and for each module we have the ancestral trajectory of shape changes, which look like this. And because it's evolving by heterochrony, we know that the dissonant adult shape has to lie somewhere on the trajectory. It can be before the ancestral adult stage, which would be pedomorphosis, or we can extend it beyond, which would be paramorphosis. So that's the set of constraints. So that's just a quick example for one of the modules. So you have that empirical ancestral dispersion around the ancestral trajectory of shape changes with the adult of the ancestor. And for instance, one possible shape for that module, which is within that empirical dispersion, so it's assumed that it's heterochronally possible. And if it's possible, then we just look at the amount of, like, the shift along the trajectory, and that's our measure of accessibility for that module. Because we have 3 modules, we need to combine all this information to assess the overall accessibility. And once we have this information for all these points on the sampling grid, then we can depict the morphospace with lines of equal accessibility, a bit like a topographic map. So that's what it looks like. So the green area corresponds to the domain of the heterochronically possible. So if it's white here, it means that you can't access it using the set of constraints that we have imposed on the system. And you see that these lines are not distributed as circles, but strongly biased. So there's important anisotropy in the space. And it means that there's discordance between shape distance, as you could measure them in the space, and actual accessibility. So despite, like, the distance between these 2 guys is the same as between these 2 guys, you see that you have to cross many more lines in that direction. So it requires more evolutionary developmental changes. It's been discussed in the literature that, of course, all the type of heterochrony do not necessarily have the same frequencies, depending on groups, depending on structure looking at. So there are basically 2 outputs, as I mentioned before. It could be pedomorphosis if your descendant look like a juvenile, or it could be paramorphosis if the descendant look like a hyper-adult. So by using the case study before, we have different parts, and we know from the case study that one module is affected more by paramorphosis, while the 2 others are affected by pedomorphosis. So if I included this additional information, so basically we have now an equal likelihood of the different type of heterochrony that give us a slightly different pattern. As expected, the real dissonant palmari is among the realm of the most accessible part of the space. And you see, again, strong discordances, and the directionality it sort of relates to the notion of directionality of evolutionary transition, because it's much easier to go in that direction for a given amount of shape changes, of developmental changes. So it doesn't mean you can't go left, but it requires much more changes of the developmental trajectories. So with this asymmetry of transition probabilities, we can now ask, once you've reached that dissonant, how easy it is to get back to the ancestral shape. So that's the observed transition in the fossil. And you see that if the set of constraints is sustained, which is the likely, the normal expectation for constraint, they are deep time concept, you see that it's much harder to go back to the opposite direction. You have to cross many more lines in terms of accessibility. So that relates to the notion of relative irreversibility. And if it's maintained during long time scale, it can generate evolutionary trends, even in the absence of directional selection. So all this stuff has some implications for morphospace analysis as we do it, and I'm going to review just a few examples. But the space, instead of well, it should not be envisioned as something really homogeneous, regular, and symmetric. Instead, that's something which is highly structured with a threshold effect and local set of rules that change depending on location in the space. So an example of implication for morphospace and disparity analysis. If you look at these 2 distribution of dots, they're actually identical, just rotated by 90 degrees. But when mapped onto the underlying structure of the space, we see that this is actually much easier to generate from an evolutionary viewpoint than this type of distribution. So geometrically, the disparity of the 2 sets of the 2 examples are identical, but in terms of evolution, that's a completely different meaning. Another type of analysis is the comparative studies of evolutionary trajectories. So it's been proposed to look at, say, the endpoints of evolutionary trajectories of 2 lineages, or we can have also different step transition, and to compare the magnitude and direction of these lineages. Well, this type of analysis rely on the fact that the space has to be isotropic, but if, as it's often not the case, the space is anisotropic, then all this notion of distance and direction can be extremely misleading. And finally, something which is quite popular at the moment in morphospace analysis when plotting like what we call phylomorphospace, so phylogeny mapped onto the morphospace, implies reconstructing ancestral character states and node. And many approaches like square change parsimony or simple likelihood model based on Brownian motion assume again the morphospace to be isotropic. So if the space is structured, that gives us quite a different picture. So that's just a quick simulation. So we impose that trend, like directional trend, on the space, and we see that we sample a few taxa, and that's the real ancestral nodes. If you apply square change parsimony, you see that the reconstructed ancestors are massively shift towards the main cloud of observed, and that's because square change parsimony require the internal nodes to be within the range of observed variation. So the reconstruction, again, is misleading. So as a brief summary, species are not static adult entities. And that's because phenotypic changes that we observe, even between adults, are actually mediated by development. And because of the nature of development and its properties, most morphospaces are then likely to display anisotropic structure. And it makes that our favorite, or premise that our favorite Euclidean notion of distance and direction do not cope well with such anisotropic spaces. And so it's advised that geometric measures of distance among adult phenotype, should be taken with caution because they do not properly reflect the dynamic of phenotypic evolution, simply because they actually do not account for the underlying variational mechanisms. Few perspective. So instead, we should try to devise developmentally meaningful measure of evolutionary accessibility. For paleontological study and many neontological study, we don't have access to genetic data, but that's not a big problem because the actual vast majority of developmental period is dominated by allometric growth of already formed parts, and we have strong tools to deal with allometry and characterize developmental trajectories using these methods. So in principle, operational proxies for accessibility can be devised even in fossil case study. And for instance, what could be a first step would be the use of juvenile morphospaces and allometric spaces. Thank you.